Hello, students. This is the fourth lecture on microbiology for medical students. And the topic of it is bacterial genetic and antibiotics. Look at the presentation. This topic, bacterial genetic and antibiotic, is rather large, so I divided into two parts. And the first part is about bacterial genetic, and second one, of course, is about antibiotics. And here you see the schedule of this lecture. And in first part, we will study this bacterial genetic. Maybe you know that the genetics is a science about heredity and variability. Characterizing bacterial heredity, I will tell you about bacterial genome, which includes the nucleoids and plasmids. And special question is the usage of specific to bacterial DNA in diagnosis. It's the PCR method. And characterizing the variability of bacteria, I tell you about phenotypic and genotypic changes in them. And the genotypic mostly includes mutations and recombinations. Um, but in this lecture, we can only study the main definitions and main principles of bacterial genetics. You know that structure responsible for heredity in bacteria, like in all cellular organism, is a DNA. And here you see the schematic diagram of its structure. It's a double-stranded, a double helix molecule. Mm, each chain composed with nucleotides, and the links between these chains in the complementary principle and the nucleic bases can pairing only in this order. The elementary unit of heredity in bacteria is a gene. The gene is a sequence of nucleotides that encodes the synthesis of specific polypeptide chain. And mostly one gene responsible for synthesis of one protein, and if bacteria possess this protein, it also has some phenotypic feature, some property. Bacterial genes may be structural if they encode the synthesis of enzymes or proteins that make up the cell organella, and other genes are regulatory. And they encode the synthesis of repressors or inductors, which affect the activity of enzymes. It's clear that repressors stop the activity, suppress it, and inductors increase the activity of enzymes. The bacterial genome means it's the all genetic information of organism. And for many bacteria, we know the structure of the genome, like in E. coli, you see here, it's chromosome, and we know in which orders all these structural and regulatory genes organized, and where is the sites for inserting of plasmids or temperate bacteriophages or migrative elements like transposons. It's well known now. The DNA in bacteria are very specific, so each species have their own DNA, and so we can use it in diagnosis of infection disease. The two main methods are DNA hybridization, uh, which based on the ability of DNA make a double strain structure after it's decomposed. And if two species of bacteria closely relative one to another, the DNA have complementary sides, and if we mix the DNA of these two bacteria, it restores a double strain structure. If they are not related, no this uh, double helix elements in the structure of mixed DNA. But more popular, the other method is a polymerase chain reaction, briefly PCR. To do this test, we need to detect a very specific fragment in DNA in each pathogenic bacteria, this fragment which never repeated in other microorganisms. And we can detect this um, specific DNA in clinical samples without doing microscopy or isolation of pure culture of bacteria. We just need to use these synthetic artificial fragments of DNA, which are complementary for specific sites in the DNA of pathogenic microorganisms. When we hit this double helix molecule, it loses the double structure, and these artificial fragments can attach to complementary sites. And then we add special enzyme, um, DNA-dependent DNA polymerase, and the set of necessary nucleotide, and the process of synthesis of DNA begins. And as a result, we have two molecules of DNA instead one. And this process repeated many times, 30 to 40 cycles, and the amount of DNA significantly increase, but only if it contains the complementary DNA of target bacteria. And this DNA then may be detected biochemically using special device named amplificator. The polymerase chain reaction is very popular in the diagnosis of many bacterial and viral diseases, especially if it's difficult to isolate the microorganisms from clinical samples. And it's very, very sensitive. The structures containing DNA in bacteria is the bacterial chromosome, also named nucleoid. Bacteria have only one chromosome, it's a circular DNA molecule, and it's rather large. In this electronic microgram, you see released DNA which released from bacteria. Bacterial chromosome longer than 1 millimeter, so we can see it in naked eyes. It 1,000 times longer than bacterial cell and contains approximately 4 million pairs of nucleic bases. And the content of DNA is rather high in bacteria. It's approximately 10% of cell volume. Bacteria have only one chromosome, but they also possess the extra chromosomal hereditary factors named plasmids. Plasmids are small circular DNA molecules that replicate independently on the bacterial chromosome. And then here you see electronic micrograph of this plasmid. See there, circular. Mm. And the plasmids are not necessary elements of bacterial cells. Some bacteria have one or more plasmids, and some don't have any of them. And, here you see the bacterial cell schematic diagram. Its chromosome, the large circular molecule of DNA, and small circular molecule, its plasmid, extra chromosomal hereditary factors. Um, Plasmids perform the different functions in bacterial cell, and here you see the list of main types of plasmids. They are F plasmids. F means fertility factor. It's a conjugative plasmid, so it can be transferred from one cell to another by the process of conjugation. We'll tell about it a bit later. And if bacteria have this F factor, it also produces special structures, F pili or sex pili, to transmit the DNA to other bacteria. A very important plasmid are R plasmid. R means resistance. It's a factor of resistance for antibiotics and other antimicrobial substances. And we return to this type of plasmids in the second part of lecture devoted to antibiotics. Some bacteria possess this called plasmids, which encode the synthesis of bacteriocins. Bacteriocins is the low molecule proteins which suppress the growth of other bacteria. Uh, the name cold plasmid originates from the bacteria E. coli. These bacteria normally live in intestinal tract of people, and it produces the substances, bacteriocins, which kill other enteric bacteria, pathogenic and occasionally pathogenic. They um, can suppress the growth of Salmonella and Shigella, Bacteroides, Enterobacter, and some others. Mhm. Pathogenic properties of bacteria also may be associated with the presence of plasmids, like N plasmid, which is responsible for production of enterotoxin affecting the intestinal tract of people, or HLY plasmid, encoding the hemolysin, the substance which can cause the hemolysis, the lysis of erythrocytes. And the non-pathogenic free-living bacteria often possess the degradative plasmids, which allows them to ingest some unusual substances like toluol or salicylic acid. So these plasmids are not necessary for bacteria, but they encode some important properties of them. Mhm. In their properties, plasmids may be conjugative, so they can be transmitted from one bacteria, from donor bacteria, for recipient bacteria. The plasmid with this property is the F plasmid, plasmid of fertility, and what is more important, the R plasmid, plasmids of resistance for antibiotic, and can be transmitted from donor cell to recipient cell. And you know that the DNA molecule is the double helix, and one thread of DNA transmitted to the mm, recipient cell, but others stay in um, donor cell. And as a result, both cells possess this plasmid. And due to this process, our plasmids, the plasmids of antibiotic resistance, um, spread very fast in bacterial populations. Non-conjugative plasmids cannot be transmitted from donor cell to recipient cell, and they're transmitted only by cell division from generation to generation, from mother cell to daughter cells. Um, some plasmids can be integrated, inserted into bacterial chromosome. There are some complementary sites in the DNA of plasmid and DNA of chromosome, and if plasmid is near the chromosome, they connect one to another. Then a special enzyme restrictase cut the DNA of plasmid and the chromosome, and other enzyme link them, enzyme ligase, link these fragments of DNA, and the plasmids became the part of chromosome. And this process is reversible. In some occasions, it is a loop of the chromosome, and then enzyme restrictase cut away this plasmid, and it became independent. And you see that the behavior of integrative plasmids are very similar to temperate phages, which also can insert their DNA into bacterial chromosome. And maybe that this kind of phages originate from integrative plasmid, from these cellular elements. And other plasmids are not integrative, they exist only in free state. The example of integrative plasmid is a F plasmid. R plasmid mostly non-integrative. And what is the two essential function of genetic material in microorganisms? It's a replication and expression. Mhm. Replication in bacteria occurs in this level. The information transmitted from DNA to DNA in the process of its synthesis. Mm. This process also takes place in DNA genome viruses. In RNA genome viruses, the replication occurs at the level of RNA with the participation of enzyme RNA-dependent RNA polymerase, and RNA serves as a template for synthesis of new molecules, new copies of viral genome. Some group of RNA genome viruses, it's the retroviruses, mm, for their replication use other strategy. They possess the enzyme RNA-dependent DNA polymerase, which can do the reverse transcription, and firstly they produce DNA template for synthesis of genomic RNA of these viruses. And some pathogenic microorganisms like prions have no nucleic acids. They name the infection proteins, and the genetic transfer is possible in this level from protein to protein. Actually, the prion proteins encoded with cellular DNA, but they change their tertiary structure after the contact with infection proteins, um, this prion, and also became prions. And what is expression? It is a process of synthesis of proteins and the information in expression of genes transferred from DNA to RNA due to transcription, and then from RNA to proteins due to translation. And if bacteria possess some protein, mhm, it acquires some uh, property. Expression of specific genetic material under particular set of growth conditions determines some characteristic of organisms, its phenotype. For bacteria, the phenotype is the morphological, cultural, biochemical, antigenic, and pathogenic properties, which we can observe under microscope or in petri dish when we cultivate them, or doing some serologic test, or um, in test with laboratory animals. In eukaryotic microorganisms, only vertical transfer of DNA is possible due to cell division from mother cell to daughter cell. But in bacteria, uh, there are two ways of genetic transfer: vertical by cell division and horizontal transfer between bacteria of the same generation as a result of conjugation, transduction, and transformation. Mm. And we will tell about these processes a bit later. Mhm. And now we go to characteristic of variability of microorganisms that may be phenotypic and genotypic. Phenotypic variability is inheritable. It means that the phenotype, its property of bacteria, changes, but the genotype stays the same. The mechanism of phenotypic variability is the repression or induction of certain genes under influence of environmental conditions. And what's important, this phenotypic variabilities appears in overwhelming majority of cells in population under external condition, and they're reversible. Mm. And there are a lot of examples of phenotypic variability in bacteria. It may be morphological changes if we cultivate when them in the presence of antibiotics, especially antibiotics affecting the production of cell wall, production of peptidoglycan. Under this condition, the rod-shaped bacteria may become spherical. But if we re-inoculate this changed microorganism to the fresh nutrition media without antibiotics, they restore their normal um, morphology in some time. Some pathogenic bacteria produce pigment only in cultivation in the sunlight because pigment source for protection them from ultraviolet. But if we cultivate the same bacteria in darkness, they produce non-pigmented colonies. Um, Capsula in bacteria also maybe change their presence as a phenotypic variability. Some pathogenic microorganisms produce capsula only in susceptible microorganisms because capsula is necessary for them as a protection against phagocytosis. But when we cultivate the same bacteria in nutrition media where there's no phagocytosis, they don't produce capsula. Mm. And it's necessary to know Which phenotypic properties do the pathogenic bacteria demonstrate in different conditions necessary for accurate microbiological diagnosis? Mm, And the bacteria also have a genotypic mutations, including mutations and recombinations. Mutations is a stable heritable changes in genomic material in sequence of nucleotides and according their role for bacteria. They may be harmful, even lethal, cause a death in bacteria. Very rare, they're helpful, and mutations may be silent. When genotype is changed, the sequence of nucleotide changes, but the phenotype stays the same in some conditions. And in this part of slide, you see the classification of mutations. According to their scale, they may point, the small mutations or chromosomal, according to their origin, they may be spontaneous or induced, and according to their direction, they may be forward, reverse, or suppressor mutations. And it is a small part of your independent work. Please find the definitions of all these kinds of mutation, and we will discuss it in practical classes. Topic, next topic, antibiotics and bacterial genetics. And what is the medical importance of bacteria with mutations, mutant bacteria? The most strains used to produce a live vaccine or spontaneous mutants with reduced virulence and high immunogenicity. Spontaneous means that they will be found in natural populations of bacteria, not undergo the influence of physical or chemical mutagens. Industrial strains, um, which used for production of vitamins, amino acids, antibiotics, are mutants with altered biosynthesis regulation. In these bacteria, mutations are in regulatory genes. Usually, they have normal genes effector and spoiled gene um, suppressor. So they produce vitamins or amino acids much more than it's necessary for their normal metabolism, and they release the excessive amount of these biological active substances, and then we can extract them, these substances from uh, nutrition media, from cultivation liquid, and use as a medical preparation. In the genetic engineering studies, the very special type of mutants are used as a named conditionally lethal. They die at temperature of 37 degrees, so they cannot multiply in human body. Why it's necessary to use this kind of bacteria? When we invade into genome of microorganisms, the sequences may be unpredictable. So for the safety, it's recommended to use only these mutants, which cannot be pathogenic for people. Mm. And oxotrophic mutants may be used as a biosensors. I think you remember from the topic of physiology of bacteria what are the oxotrophs. Oxotrophs is bacteria which need some substances for their growth. And using the property of these mutants, we can detect the presence of some chemicals in clinical or environmental samples. If mutants can grow in this substrate, so the substance present. If no growth, no um, presence of this chemical. And this method is sometimes more sensitive than biochemical methods. And other kind of variability in bacteria is a genetic recombination. In eukaryotic microorganisms, the recombinations mostly associated with sexual multiplication because each new generation takes one part of genes from mother and one half from father. And in new generation, we have new combinations of these genes which are important for evolution of eukaryotic microorganisms. But bacteria multiply only asexually, and the genetic combination in them different from eukaryotes. It's only partial integration of the genomes of donor bacteria and recipient bacteria. So you see also two bacteria participate in this process, donor and recipient. Donor gives its DNA, recipient takes it and includes into its genome. Recipient bacteria can include the linear fragment of donor DNA, or what is more usual for bacteria, the circular fragment of the donor DNA. And there are some ways in which donor DNA can be transported into recipient cell. The simplest one is the transformation. In transformation, the genes are transferred from one bacteria to another as a naked DNA without carriers or cell contact. The bacteria donor die and undergo the lysis, its DNA released, and bacteria recipient absorb this fragment of DNA and include in its chromosome. This phenomenon was investigated in the research with bacteria pneumococci. Pneumococci produce different strains, some of them non-capsulated and non-virulent, produce rough colonies, and if we inject them into laboratory animals, the animals stay alive. Bacteria produce smooth colonies. They possess a capsula. Capsula is important pathogenicity factor. And if we inject these bacteria to laboratory animal, they die fast. If we kill these capsulated microorganisms with heating and inject into mouse, stay alive because that bacteria cannot cause harm it. But if we mix this harmless live bacteria without capsula and this also harmless killed bacteria possessing capsula and inject this mixture to laboratory animal, this animal die, and in tissues of this uh, dead mouse we can find live capsulated pneumococci because they acquire this property capsule formation with the DNA released from donor cell. Other way of genetic transfer is a conjugation. Transfer of genetic information via direct cell-to-cell contact, and this process mediated with fertility factor. The donor cell must possess F plasmid or F plasmid integrated into chromosome and named HFR. And here you see the electronic micrograph of process of conjugation. This is a donor bacteria possessing F plasmid, and if it has F plasmid, it also produces F pilus or sex pili to transfer the DNA to the recipient cell. In conjugation, chromosomal genes or plasmid can be transmitted, and here we see the transmission of F plasmid, double-strand DNA, and only one copy of DNA transferred into the recipient cell, and as a result, both cells possess this plasmid. In this way, F plasmid or R plasmid may be transmitted from donor cell to bacteria, to recipient bacteria. The F factor, F plasmid, is the integrative plasmid, so it can insert itself into bacterial chromosome and turn into HFR factor. HFR means high frequency of recombination. And this bacteria also produces sex pili to contact with recipient bacteria, and it's possible to transfer the chromosomal genes, also only one thread of DNA from donor cell to recipient cell. If this contact lasts only a few minutes, one or two genes may be transferred from donor to recipient. If contact is prolonged, many genes of recipient of donor can be transmitted to the recipient and then included into its chromosome. And the frequency of common transmission of some genes were used to reconstruct the bacterial genome, the sequence of gene in the bacterial chromosome. If some genes is located near one to another, they transfer it together. And the last way in which bacteria can transfer the DNA from donor to recipient is the transduction. And the bacteriophage is a virus of bacteria participate in this process. The bacteriophage infect the donor cell, multiply in it, and this infection leads to uh, destruction of bacterial chromosome. And in the process of maturation of new bacteriophages, most of them, of course, possess normal phage DNA, but some of them may take the fragment of bacterial DNA. And this donor cell undergoes a lysis, it's die, and the new bacteriophage is released and infect new cell, the recipient cell, but this bacteriophage containing bacterial DNA, it's defective. It can attach to the bacterial cell, inject this DNA, but this DNA not viral. So the process of production of new virions cannot be take place in this bacteria, and this donor DNA is included into chromosome of bacteria recipient. These bacteria stay alive, they multiply by binary fission, and the gene of donor bacteria transferred to the progeny of this recipient bacteria. And bacterial genetic is very important for development of genetic engineering. Genetic engineering is the design of organisms with specific proteins. It's the introduction of foreign DNA, it may be human DNA or viral DNA, into bacterial genome, sometimes in viral genome also, to obtain high molecular biologically active substances. For example, genetic engineering used for production of insulin, uh, the human hormone, um, which is necessary for treatment of diabetes uh, type 1. The insulin is a protein, so in human cell is a gene responsible for its synthesis. It's possible to take this gene from human cell and then we need to transport it into bacteria. How can we do it? We need some vector. Vector is a plasmid, um, conjugative plasmid. Mostly we use R plasmid, plasmid of antibiotic resistance with this genetic marker of resistance for some antibiotic. And we insert human gene into this plasmid. Then using enzyme DNA-dependent DNA polymerase, we obtain many copies of this plasmid and mix it with bacterial culture. And some bacteria became recipient and then take this plasmid from nutrition media. And in the next step, we need to differentiate this recombinant bacteria from other bacteria without this recombinant plasmid. How can we do it? Remember that this plasmid contains marker of antibiotic resistance. So we add antibiotic and all these bacteria which have no recombinant plasmid, they die. Only this bacteria survive, it multiply, and all its progeny possess this plasmid with human gene, and all these bacteria start to produce human insulin, which used as a medical preparation. And students, this is the end of first part of this lecture, and look second part, which is devoted to antibiotics.